DNA synthesis. We're creating an identical DNA molecule to our original. Well, we have 46 chromosomes and they're becoming double-stranded. Keep in mind, an interphase, it's uncondensed in reality. And I think we'll have time. We're going to do a warm-up that sort of reviews this concept. But yes, we're duplicating all of the DNA in our genome. The genome refers to the entirety of your DNA inside the nucleus of your cells. All the genes, all the rest of the DNA, which really is just sometimes called junk DNA because it doesn't have any, it has other functions that you might explore in other classes, but okay, so let's see, what else? Apoptosis, I wanted to talk about again. So this, after S phase, right, after your DNA is copied, 3 billion bases are copied, 3 billion nucleotides to create another DNA molecule. That's a lot. And so errors happen, and like maybe, you know, you'll have a mismatch, like you have a base that's not the same on accident, and we have enzymes that can correct for that. But those are easy. If the extent of the damage or the error is so large that it can't be corrected by these mechanisms, then the cell undergoes apoptosis, which we talked about last time. This is programmed cell death. Programmed cell death, it's an intentional cellular self-destruction to prevent this cell from dividing, essentially, because these errors can also often lead to cancer, to cells that are cancerous. So the cell has mechanisms to destroy itself to prevent that. Of course, cancer cells evade these mechanisms and they're still able to overcome apoptosis and divide, but most of the time they will do this. And so apoptosis, I just wanted to talk about a little bit because it's so interesting. Just really quickly, I wanted to show you that it's really useful, this cell self-destruction mechanism. And so I'm going to share my screen with you. I was doing some Khan Academy reading, and so one of the most important roles of apoptosis is in development. And so our hands, or anything with digits, hands and feet, actually all vertebrate animals with digits, will have this sort of web of tissue of cells, but to form the digits, you can see that in between these cells actually apoptose and they die off to form your fingers. So that happened when you were an embryo. It's pretty amazing. There are a couple other important mechanisms. So number 3 on the Halloween extra credit quiz deals with coronavirus, just like a funny question. And so just unfixable DNA damage, we talked about that. I just mentioned that, right? This cell will apoptose. But another thing that cells will apoptose is if they're infected by a virus. I'm trying to find it. So cells infected by a virus, right? So the virus can die with the cell. It's kind of amazing that we've evolved to do this. Does radiation stop them from dividing or growing? Yes. So radiation is toxic and it will kill cells. So that's the key. That's why radiation treatment. But the problem with radiation treatment is it's going to kill normal cells too. which is often why it's such a, harmful thing. Yeah, if you think about kind of like Chernobyl, like that kind of high level radiation is so deadly. It just obliterates your tissues. It's really bad. That's why they do it for cancer, but it's kind of a last resort thing because it obliterates all your tissue, not just the cancer. But okay, so that's another, and then there's a couple other things with apoptosis, but those are the two sort of main. This is an important process. That's all. Okay, so let's, besides the extra credit quiz, are there any questions before we start to talk about mitosis today? Cell division by mitosis? Okie dokie. So let's go back. Now, do I have my... Oh, I have a joke for you guys. What did one cell say to the other after stepping on her toe? Nice. You've heard this one. Don't step on my toes, sis. Yeah, isn't that funny? All right. Okay, moving on. So actually, this is sort of a warm-up I mentioned. I want to kind of review this idea of DNA replication, kind of practice what we've been discussing. So human body cell has 46 chromosomes. How many chromosomes are present after DNA replication? How many sister chromatids are present? So I'll give you a minute to work on that, and we'll talk about it. Okay, I'll give you one more minute, then we'll talk about it together. It's kind of a good segue for mitosis today. Did I hit record? Oh, I did. Okay, just checking. All right. Anyone still need time, or you want to talk about it? So who wants to tell me how many chromosomes are present after DNA replication? We start with 46 chromosomes. How many do we have after we duplicate all of our chromosomes during DNA replication? Ooh, I fooled you guys. Oh, we got some. Yeah. Okay, so it was a trick question. I'm glad I did this as the warm-up. It's a tough concept, so you know, this took me a long time to wrap my head around. It's 46. Yeah, it's still 46 chromosomes, right? The only difference is that we're going from single-stranded to double-stranded. Is that ringing any bells? With those sister chromatids, those identical sister chromatids. Now, just keep in your mind though, DNA replication happens during S phase, right, of the cell cycle. And this is during interphase. Meaning in reality, it's uncondensed, right? So the cell, the 46 single-stranded chromosomes that are going to be duplicated, they're uncondensed, right? So it's like a bowl of spaghetti. And then during S phase, when they duplicate, it's just this long uncondensed bowl of spaghetti that becomes a little more, I guess a little more, I don't know how to say it, more spaghetti. So it's kind of like this. Becomes a bit more intense, I guess is the word. More dark and concentrated because you duplicate the entire length of that DNA, right? I illustrate it like this, however, just to show you that that's what's going on. And if you were to condense the chromosomes, they'd look like this here and this after S phase. But that brings us to our topic of today, mitosis. The first phase of mitosis we're going to talk about, the chromosomes, or I'm sorry, the chromatin that's uncondensed condenses into chromosomes. And because they've been duplicated, during S phase, now that we enter M phase, mitosis, we are going to condense our chromatin and form these double-stranded chromosomes. So this is what we're going to talk about today. This cell dividing into two. And you're going to see that after this division, the chromosomes will be single-stranded again. You're actually going to see exactly how that is. And even how you start with 46 chromosomes and end with 46 chromosomes in each new cell, but they're single-stranded again. We're going to look at that in detail. And of course, I forgot to answer the question, how many chromatids are present after DNA replication? Perfect, 92. So 92 chromatids, sister chromatids to be exact. That's what we call them. And you only have these chromatids with double-stranded chromosomes, right? Okay. All right, let's do it. Let's do it. You guys got this? Okay. So if you look at the cell cycle, which were talking about last time, we are entering the mitotic phase after G2 of interphase. And before we do, I'm just going to show you what the cell looks like again, just up towards the top here. What it looks like in interphase, and then what it'll look like when we enter mitosis. So we're doing mitosis. And I just want you to recall or have this in your notes. In interphase, our cell looks like this. So in G2, it looks like this, uncondensed chromosomes in this chromatin state. It's our spaghetti. And then the first phase of mitosis is called prophase. I'm just going to draw a big cell. Whoa, I drew a really good circle. Wow. Okay, and there's going to be a couple key events that happens during prophase. Problem with my circle is I made it kind of too big, but that's okay. I'll fit it. Fit the. Okay, I'll write prophase here. This is our first phase of mitosis. And there's going to be a couple key events that occur. The first, namely, the chromatin is going to condense into individual chromosomes. Okay. So let's write that. Let's write that. So during prophase, chromatin, gosh, sorry. Chromatin condenses. It's going to become tightly wound up, packed into chromosomes. Condenses into chromosomes. So actually what's really interesting is our 3 billion nucleotides of DNA, if it were all stretched out from the cell, if we all just kind of like stretched it all out, I think it's like the statistic is it goes 7 feet. So to get it into one nucleus of one microscopic cell, it has to be really condensed, packed in there. So during prophase, chromatin condenses into chromosomes, individual chromosomes, of which we have 46, right? And of course, that's too many to kind of draw through mitosis. So I'm going to do 4 just to show you what's going on. But before I draw them, there's one other thing that I want to mention because I'm going to demonstrate it here. But the other thing that happens during prophase is the nuclear membrane or the nuclear, I'll write the nuclear envelope or membrane begins to break down or dissolve. And so I'm going to visually represent that as this nucleus that's fragmented, starting to be broken apart. Okay. Now, as we talked about the last few lectures, what was I saying? Oh, these, a lot of these cells that go through mitosis actually really well, I mentioned it, the somatic cells, right? The somatic cells are doing mitosis and, right? And we talked about the reasons, growth and development, asexual reproduction, and repair and regeneration. Now, we have 46 chromosomes, 2 sets of 23 in all of our somatic cells. Does anyone remember what that's called when we have 2 sets of chromosomes? A cell or an organism that's comprised of cells with 2 sets. Diploid, that's correct. So this cell is diploid because it's going to have 2 sets of chromosomes. So it's 2n equals 46, right? That's the diploid number. N is representing how many chromosomes are in one of those sets. Now, since I'm not going to draw 46 chromosomes, I couldn't even fit them in this picture, we're going to do 4 for this example. So we have a diploid 2n equals 4. So we're going to draw 4 chromosomes. I hope you have your colors. The next few lectures, you're going to want especially blue and red. But I'm going to do 4 chromosomes, 2 sets of 2, 2 homologous pairs. And they're condensed at this point, so I'm going to do, let's do 1, 2. Those are my chromosomes. Okay. So this is prophase. Nuclear membrane is breaking down. Our chromatin uncondensed condenses into chromosomes. That's the first step of mitosis. The second is called metaphase. And by this point, the nucleus has completely dissolved, goes away. And we have another event that's going to occur during metaphase of mitosis. Something I actually forgot to draw in, but G2 prophase, at some point, scientists aren't exactly sure when, but at some point in this transition, there's going to be what is called a spindle apparatus, or a spindle fibers begin to form. And they kind of look like this. They're composed of these proteins that are going to act as basically, they act as strings, like kind of like a puppet on a string. And they are going to move our chromosomes around. These spindle fibers, they attach to our chromosomes and move them around during mitosis. But if you want to add to your prophase drawing that these are present on either end, go ahead, because they form. I totally forgot to write them in, draw them in. But what these spindle fibers are going to do, they're going to, like I said, actually elongate and attach to our chromosomes. And during metaphase, they're going to move them to the center. No. This is metaphase now. I just mentioned that these spindle complexes form during prophase, or G2, late G2. So I forgot to write them in before, but now they're here. And what they're doing is they center the chromosomes during metaphase. in the middle of the cell. Single file. So I'll show you what that will look like. Okay, so during metaphase, chromosomes align single file in the center of the cell, or the middle. To help you remember, middle, metaphase. So they're all in the center here. Center of the cell. And they're moved there by these spindle fibers. Yes, we're in metaphase. So in metaphase, the chromosomes are being moved by these spindle fibers that attach to the chromosomes at the centromere region in the center, and move them in the center of the cell, the middle. That's metaphase. So this is metaphase, what happens during metaphase. I'm going to underline single file because when we talk about meiosis, the next form of cell division, next week, it's similar to mitosis in a lot of ways, but also different. And this single file thing is important as a difference. So during mitosis, right, they're going to align single file one by one here during metaphase. Okay. Questions? Other questions? Wow, I just realized, I made such a big mistake. Oh, wow, I've never done this. I am so sorry. Does anyone see it? I don't know why I forgot this. I guess because I'm human, but I knew something was off. Does anyone know what I'm doing wrong here? It's an easy fix, but it has to do with the chromosomes. Maybe it'll have learning opportunity. Yeah, it'll help you remember it. Help me remember not to do this again. They're not double-stranded. Yeah. So we enter mitosis after S phase. I took so much time to talk about that and I didn't even do it. I drew them single-stranded, but this isn't how we start mitosis. So if you want, I can redraw prophase. I might just start over. Let's just start over and go through it quickly. You already have the notes for prophase. But yeah, I apologize about that. But this is what it looks like. Can't believe it. I'm like, how is this gonna, oh boy. Okay, so prophase, right? It's like, I knew I didn't draw it like this before. Something was off, but it's gonna look like this. They're X's. I'll give you guys time if you want to redraw or add sister chromatids to the ones you did. But that's what it looks like. Gosh, sorry. So this is a note for metaphase, right? But during prophase, our nucleus is dissolving, the membrane surrounding the nucleus is dissolving, chromosomes condense into double-stranded, chromatin condenses into double-stranded chromosomes. Jeez, come on, Ryan. And it's diploid 2n equals 4. In reality, we have 46 X's, okay? 46 double-stranded chromosomes. They all duplicated during S phase. Okay, let's move on. Back to metaphase. Of course, forgot the spindles again. Okay. There we go. That looks better. Here's metaphase now. Okay. They're in the center or middle, metaphase middle. The next phase is called anaphase. During anaphase, sister chromatids are pulled to opposite ends of the cell. And for your notes, if you want the name of these structures that have these spindle fibers emerging. So these are mitotic spindle or spindle fibers. So the spindle fibers are actually going to pull apart the chromosomes. They're going to pull the sister chromatids apart from one another towards opposite ends of the cell. okay? And I'll show you what that looks like. Okay, the red one above. Oh boy, come on. Usually I do it multiple circles, but save time here. Okay, so this is Anaphase. Spindle fibers are pulling the sister chromatids apart from one another to opposite ends of the cell. Okay, is everyone caught up here? Drawing separate cells. Usually I have giant whiteboards, so I do like cells one after another. Obviously I can't really do that right now, and I don't want to, you know, well, guess I could, but it wouldn't, no point. All right, one more phase to go. So I actually am going to redraw the cell because it looks slightly different in this final phase. The last phase is called Telophase. And during telophase, the cell elongates and starts to pinch in a little bit, like that. And nuclei, nucleus, plural nuclei, two nuclei begin to form at opposite ends of the cell, like that. So let's start to write some of this down. So the cell elongates, begins to pinch in the center there. And nuclei form. at opposite ends. And after we pulled apart during anaphase, they're going to look like this. Florian, that's correct. Yeah, exactly. So they're back to single-stranded. Our sister chromatids of the double-stranded chromosomes get pulled apart during anaphase. And so at either end of the cell, you're going to have those, what used to be the sister chromatids attached to one another, separate chromosomes now. And to sort of emphasize that point, the last stage here of the cell cycle, and it's always it's almost always coupled with when you talk about mitosis, is cytokinesis, which is the division of the cytoplasm. So when the cytoplasm here of our elongated cell starts to pinch in, it pinches in all the way, kind of like if you have a water balloon and a string tied around it, and you start to pull it tighter. It's a contractile ring that contracts and actually pinches the cells into two. And this is called cytokinesis. So our cell has pinched into two brand new cells. And at this point, the two nuclei have fully formed their membranes. And we just did cell division. But for the cytokinesis, I'm going to write the division of the cytoplasm. And quite remarkably, each one of these cells is diploid. 2n equals 4. The reason that is, if you remember, a chromosome refers to a unique set of information. Each chromosome in this cell is different from the other. Each chromosome in this cell is different from the other. So if you imagine in a human cell that's going to divide by mitosis, we started as 2n equals 46. 46 chromosomes that are double-stranded, meaning that sister chromatids are identical, and you have a unique 46 unique chromosomes, right? At the end of mitosis, you're going to retain all of that information. You just go from double-stranded to single-stranded in each of your cells. The sister chromatids are identical that come apart. And so you still have all of the same amount of information in each new cell. That's the key here. Oh, what is the word? Division, yeah. So division of the cytoplasm. The cytoplasm divides into two. So each of these new cells still has 4 sets of information. Pretty amazing. Okay, what time is it? So the spindle fibers during telophase, well, they're kind of done being important in telophase. They don't do anything. I'm pretty sure they just disassemble. They disassemble and the components are recycled, or the proteins will become part of the cytoskeleton. I actually don't know specifically, but they definitely go away because they have to assemble again at some point. So I'm guessing they break down and depolymerize. That's a good question though. They are paired. Okay, so they are paired during cytokinesis. No, they're at separate ends at this point. Anaphase has pulled them apart. I think that's what you're asking. What was under a star during telophase? The cell elongates, one cell elongates, right? We're still with one cell, and starts to pinch in, and that's correct. And then 2 nuclei form at opposite ends, right? They were starting to form for that elongated cell. In each new cell are the chromosomes then paired. Oh, good question. Yes. Yeah. Remember, we have 23 homologous pairs. It's actually a great question. So let me show you what each of these cells looks like in a human after mitosis. There it is. So they're still paired, right? So really important concept. At the beginning of mitosis, we have 46 X's to separate. At the end, we have 46, in each new cell, we have 46 single-stranded chromosomes. Still 23 pairs, but these 23 pairs are single-stranded versus the 23 pairs being X's double-stranded, right? At the beginning of mitosis. 46 X's versus 46 noodles. Great question, yeah. So they're still paired, but they're just single-stranded pairs now, homologous pairs. And I tried to show you that kind of like these are homologous pairs, and they're the same length, right? One's from mom, one's from dad. I just drew 2 sets of two pairs, when in reality we have 23. Because that would take all night and a much bigger whiteboard. Other questions? Great questions. Jeez, it's awesome. I love it. You guys are thinking. It's amazing. Good on time. Notes here. All right. So let's see where we get. 20 minutes. So now what I like to do is a sort of review of sorts. And then an introduction, if we have time. Yeah, we'll see where we get. But I like to do this big chart that reviews mitosis and the concepts and key aspects of mitosis. And then it's going to compare it and contrast it to meiosis, which we're going to do next time. And it's a perfect sort of lead-in for next time and a review of this lecture this time. Questions. Is circular. Cytokinesis is kind of like a, yeah, I would think of a water balloon. That's my favorite example to use, where if you have a water balloon and you have a string around it and you tie the string tighter and tighter, the water balloon starts to pinch in the middle and then it will separate into two kind of thing. That's kind of what's going on. Just trying to draw it out. Honestly, all you need to really do for cytokinesis is just draw a line down the middle. It's the splitting event of the cytoplasm. Yeah, that's all you need to know. Just in reality, what's going on is there's this ring of fibers that contract and squeeze the cells or the big cell and pinches it off. Okay, so let's do this chart. It's like my favorite chart. It's going to be a mitosis meiosis chart. And I learned with my other school, did this lecture the other day, that I don't have enough room to do both mitosis and meiosis. So I'm going to do mitosis first, and then we'll erase it and do my meiosis depending on time. But what I'm going to do, mitosis, I'm going to do the who, the what, the where, the when, the why, and the how. So who, what, where, when, why, and how, which we just did. We did the how, but we'll get there. So this is sort of a review of last lecture and this lecture, right? So the who, what organisms do mitosis? Who's doing mitosis? Almost all cells do mitosis. Almost all cells. So animals, plants, fungi, bacteria, a kingdom known as protists. These are like amoebas and plankton and stuff. So really everything on the planet does mitosis. Like 99.9%. There's like a few exceptions. They do meiosis, but okay, so what's going on? As we just saw, one diploid cell. So this is 2N, right? Becomes two diploid cells. Okay, as we mentioned, somatic cells, this is where this is going on. Somatic cells do mitosis. So body cells, right? These were our skin cells, intestinal cells, and then the somatic cells, when you grow and develop. I guess the point being a lot of cells do mitosis. Okay, speaking of growth and development, well, for the when, I'm gonna write continuously, right? Cells in you and me and most life on the planet is constantly doing mitosis, so continuously, but as I mentioned, only some divide during development, right? Like muscle cells, brain cells, things like this, they're not constantly dividing or else, you know, can become cancerous. So they stop. So they only divide, so some only divide during development. So some only during development. Okay, the why. This is what we talked about last time, right? Growth, repair, regeneration, and asexual reproduction. Ran out of room for how, but I can fit it in. We just did it, right? We just went through how a cell divides mitotically. I'm going to abbreviate it. Oftentimes, biologists abbreviate it as PMAT, Prophase, Metaphase, Anaphase, Telophase. PMAT. And cytokinesis, if you want to add that in as well. PMAT plus cytokinesis. Cytokinesis. Okay. Well, let's see here. Trying to decide if I want to do meiosis for all of this now or next time. Let me see here. Yeah, let's do it. At least start it. And if we don't finish, we can finish next time. Okay. Now we're going to go through these for meiosis. So we're kind of departing from mitosis. We're going to look at our next type of cell division, which we've mentioned before. And it's called, I'll do it in, let's do it in purple, meiosis. We're going to do same questions. So we're comparing it, but for meiosis now.